Welcome to today's live broadcast titled Experimental Design and the Reproducibility Crisis, Pitfalls, Power, and Practical Best Practice. It will be co-presented by Associate Professors Dr. Joe Garner and Dr. Brianna Gaskill. My name is Megan LaFollette, and I am the 3Rs Fellow for the North American 3Rs Collaborative, and I'll be your moderator today. Today's seminar is brought to you by LabRoots, the New Jersey Association for Biomedical Research, the National Center for 3Rs, and the North American 3Rs Collaborative. Thank you and sponsors. For more information, please visit them on their websites. I would like to remind everyone is anticipate by submitting as many questions as you want at any time during the presentation. To do so, simply type them into the Ask a Question box and click on the Send button. Also, please notice that you can share this webinar on your personal social media. Just click on the social sharing tab to let your friends and colleagues know about today's live event. If you have trouble seeing or hearing the presentation, just click on the Support tab at the far top right of your presentation window, or report your problem via the Ask a Question box. I'd also like to note that today's presentation offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window to obtain those credits. Now I'd like to introduce our presenters today. First, we'll be hearing from Dr. Joe Garner. He is an associate professor of comparative medicine at Stanford University, and he leads Stanford's Technique Refinement and Innovation Labs. Further, he has interest in abnormal behaviors in animals and humans and the scientific impact of welfare problems in laboratory animals. Welcome, Joe.
Hi there.
Then, who's an associate professor of animal welfare at Purdue University. She leads Purdue's Laboratory Animal Welfare Research Group and has specific interests in animal welfare assessment, rodent well-being, and elucidating the scientific impact of welfare problems in animal-based research. For a complete biography on Dr. Garner and Dr. Gaskill, please visit the Biography tab at the top of your screen. Dr. Garner, you may now begin your presentation. Thank you.
Thank you, Megan. Thank you, everybody, for being here. It's a real pleasure to be here with you today. So I always like to start just with some disclosures and conflicts of interest. I serve on the Scientific Advisory Board for a number of organizations, including the TLC Foundation for Body-Focused Repetitive Behaviors, the Beautiful You MRKH Foundation, and I previously served on the Advisory Board for Tourette Syndrome of America. I consult for a variety of different companies and organizations, and I have donors both from industry and from private philanthropy. None of these disclosures are conflicts for today's presentation. So with that, I'd just like to thank you all for being here again, and let's jump into it. So I think if you are here today, in some way or other, you care about statistics. So what is that? Do you care because good experimental design is just the right thing to do? Do you care because our cooks would like better number justifications? I think we would all love to see that. Do you care because you have some idea that statistics maybe could reduce animal use or otherwise improve the 3Rs? Are you here because you care about the ethics of animal use? Are you here because science is meant to be reproducible and maybe you have a suspicion that there may be an issue there? Are you here because we'd like to improve translation to human outcomes? Again, I think that is something that we would all very much like to achieve, especially in the current environment. Are you here because efficient designs mean quicker drug development? Again, something I think that is very pertinent today. And are you here because you care about public health? Well, in fact, all of these, I think, are just different sides of the same issue. I think if you care about any one of these things, in fact, you care about all of them, even if it might not be immediately obvious. And statistics and experimental design is the lowest hanging fruit to address many of these issues. So why would I say that? Well, what I'd like to do for the next few minutes is work through the chance that a basic discovery or a basic animal project will result in a human drug. So first of all, we are in what is commonly referred to as a reproducibility crisis in biomedical science right now. And there's now a number of different data sets that show us that when we formally attempt to replicate any given basic research finding in pre-medical work, then you have somewhere between a 10% and a 50% chance of being able to do that. In other words, generally speaking, the majority of preclinical research is not reproducible in another laboratory. And Friedman and colleagues try to estimate the cost of that to just the US academic effort. And so we think there's probably about they think that there's probably about a $28 billion a year cost to the US research effort just from the non-reproducibility of experiments. And obviously, if we could fix this problem to at least some degree, it would benefit everybody enormously. So let's suppose that we've found a good lead and we've been able to reproduce it, and a drug company's taken it up and they've been able to reproduce that basic work in-house, and they begin to take the compound through the pipeline. Well, actually, only roughly 35% of compounds entering the pipeline make it to human trials. And this is generally due to issues around lead optimization, industrial chemistry, preclinical toxicology, things like that. That is probably something that is a pretty fixed number. And so the real question then is, what is the chance that basic discovery, once it gets into human trials, is actually going to make it to market? And again, there's a number of data sets looking at this, and this work, which came from Kohler and Landis in 2004, would suggest that on average, it's roughly 1 in 9 compounds actually make it through to market. And there's a great disparity between different fields of discovery in that number. And if anything, these numbers have gotten worse since 2004. So when you dig down into this, we see somewhere between 3% and 20% of drugs will make it through, depending on indication. It's very interesting that cardiovascular drugs are actually the best performing class. You will also be relieved to know in more recent data sets that vaccines are also an extremely well-performing class of compounds or interventions. So again, when you dig into these data sets, and there's American data sets, there's European data sets, you see that somewhere between 20 to 50% of these failures are due to human toxicology or other non-animal related sources of failure in the clinical trials, in the human trials. In other words, somewhere between 80% and 50% of these failures are because the drug under consideration either doesn't work or doesn't work well enough to move forward. And again, in other words, that means that somewhere between 50% and 80% of these compounds, which seem to work in animals, really are not working or not working well enough when we take them into humans. So there's a huge false discovery rate coming out of animal work into humans. And we call this problem the translation crisis. So often at this point in a talk, people think that I'm crazy. So just to give you some lovely examples, the first is a review, both of these reviews are from 2010, of the current state of play in Alzheimer's research. So this first review is looking at all published studies of the genetically modified mouse model of Alzheimer's, the APP knock-in mouse. And of over 200 plus studies of, I think over 100 compounds, if I remember correctly, none have been replicated in translational human studies. And in fact, several of those human studies actually had to be ended early because of unexpected side effects. So that's a 0% success rate. In the case of stroke, this is a very famous example. And in fact, this team, in particular, Emily Sainer, has done several follow-up studies looking at stroke. But again, 500 plus studies reporting effective treatments for ischemic stroke in animal models. only 2 of which translated to humans, and 1 of which we already knew about before the animal work was done. So in fact, only 1 of which actually translated forward from animals to humans. So that's pretty terrifying. But we can begin to sort of plug these numbers into some economic and process models of the pipeline that are out there, and we can begin to think about how many projects we might need to launch in order to actually get a drug to market that's going to help people. So using the numbers that I've shown you, when we plug in our real-world worst-case scenario with a 10% reproducibility and a 3% translation rate, the overall success rate is less than 1%, obviously. It's about 0.1%. And in fact, we would, on average, have to launch 947 projects in order to get 1 success. Now, bearing in mind that 3% is cancer, and most cancer studies involve a decent number of animals with category E procedures. As an IACUC member, and I wish we could all sort of hold up our hands and interact a little bit here, but normally when I ask an audience, how comfortable do you feel about those category procedures, knowing that you have roughly a 1 in 950 chance that this is going to make it through to humans, a lot of people could begin to feel quite squeamish, and I think that's justifiable. If we plug in some sort of average numbers that we can get, again, out of the available literature, with a 30% reproducibility rate and an 11% translation rate, then we're in a much better state. In that case, we need about 86 projects before we're going to, on average, get a success. And if we plug in our current best numbers, which is a 50% reproducibility rate, a 20% translation rate, then we are seeing that we would only need to launch, on average, 28 projects per successful translation into the human clinic. So I really think it's important to talk through these numbers, because I've definitely heard people claim that the reproducibility and translation crises aren't real, or that they're being made up or blown out of proportion by animal rights organizations, and that's simply not true. And I would really caution anybody here who thinks that that's a good argument that it's not, because these numbers are very easy to look up. It takes a very, very quick literature search on Google to find the numbers you need to recreate this table. So I did have a wonderful animation of Beaker here. This was meant to make everybody chuckle at home. And again, I can't hear you, so I don't know if it works or not. But the real key message I think that Dr. Gaskill and I want to get to you today is that this current panic is unwarranted. There's a lot of people running around with their hair on fire, freaking out that animal research is in fundamental jeopardy, and so on and so forth. And that's simply not true. We actually know what makes for a reputable study. And we actually have a very good idea of what makes for a translatable study too. And we have ways of testing and demonstrating these ideas. And so the real issue here is yes, there's a crisis in terms of what is successfully translating and what is successfully being reproduced, but it's more a crisis of culture and changing hearts and minds. It's not a crisis of not knowing what to do. It's not something fundamentally broken with science, and that is really what we're going to talk about today. So let's put some yardsticks on this. What is the highest success rate that we can expect? Well, if we had perfect reproducibility, which is probably impossible, and we had perfect translation, so if every experiment was reproducible and every compound that enters the pipeline only fails for other reasons, but every compound that enters actually works in humans, then we'd still have only a 68% translation rate. That's the best translation rate we can expect given all of the other issues in the pipeline. And that would lead us to launching maybe 4 projects on average per success. So that's our yardstick. And actually, if we plug in some reasonable numbers, so if we plug in 80% reproducibility and an 80% efficacy rate, then we're at 7 projects per success. And I don't know where you where you fall, but honestly, anywhere from that reasonable 7%, 7 projects per success to 28 projects per success. In that case, if the science is good and everything is being done to maximize the welfare of the animals involved, then as an IACUC member, I would be okay with a category E procedure. So again, I really don't think that this is sort of a nail in the coffin of biomedical research or anything like that. But it is something that I think we can improve, and it is something that I think we owe science and public health and animals to improve, and I'll explain why in a second. So where did I get these 80% numbers from? Well, if you remember, we are supposed to power our experiments to 80%. And as Brianna is going to run through, power is the chance that if a result is there, that you'll detect it. And that sort of directly translates to reproducibility. So if we're powering at 80%, we should have an 80% reproducibility rate on average. And I think we can also apply that logic to efficacy. Like if this compound really does work, there should be about an 80% chance that we can get it to work in humans. And that's where I get that 80% and 80% from there. So always explain your assumptions because they're usually questionable. So the general failure of compounds to make it through the pipeline is referred to as attrition. And attrition has been well discussed in the drug development literature for a very, very long time. This is beautiful work from Paul and colleagues, and this is the best economic model of the pipeline that I'm aware of. From this model, we know that of the roughly $2 billion it costs to bring a drug to market, roughly 75% of that cost is the cost of all of the other compounds that are going to fail in the pipeline in that company whilst that success is being developed. But only 3% of this cost is in preclinical work. So in other words, almost all of the cost of drug development is in the failure of human trials. And this leads to many of these sort of perceived societal ills of the pharma industry. And I think it's really, really important to understand this. If you are sinking this enormous amount of investment for a relatively low rate of return, you can't afford to make drugs for people who don't seek help, for rare conditions, for acute conditions. And once you begin to work through all of the different things that make an indication profitable, then it really does begin to explain a lot of the sort of development biases that you see within the industry. And that's just economics. It's not any kind of malfeasance. But it is why, for instance, we have these huge healthcare disparities in terms of the availability of drugs for different indications. And I'm happy to talk through some examples of that in questions, if it's of interest to people. So this is why I think this is also a public health crisis. I think if we can fix this problem, then we can begin to develop drugs for underserved communities. And I work with a lot of underserved disorders, so that's an issue close to my heart. We can focus more on women's health, which is a really important consequence of parts of this issue. We can afford to develop drugs for populations that maybe can't afford to pay for them. Drugs like anti-malarials, for instance. Anyway, if we try and superimpose on this pipeline the extent and the invasiveness of animal use, these are very, very hard numbers to estimate. But I think we can probably agree that the majority of the really invasive animal use is occurring in preclinical toxicology. And given that attrition may be unavoidable, right? We may always have that same total failure rate. A lot of people in the drug industry have basically argued, let's try and move this attrition into preclinical stages where it really doesn't cost anything. In other words, let's try and fail fast, fail early, and fail often. And I love this idea, because if we can do this prior to preclinical work, then everybody wins, right? So we're avoiding the business cost, we're avoiding the welfare cost, and all of that increased efficiency in terms of time, effort, and financial resources can be poured into some of these broader public health issues that I mentioned. So that's really what we're trying to get to as our long-term outcome. So how do we get there? Well, it turns out that there's been a sort of explosion of emerging literature since the mid to late 2000s, and this is just a small fraction of some of those papers. And what's really fascinating about this emerging literature is that even though most of these people didn't know each other when they were writing these papers, they're incredibly consistent in what they're saying. So it doesn't matter whether somebody's writing about stroke or about cancer or whatever the indication may be. All of these papers, in one way or another, argue that there's an inherent value to animal work, that the translation and reproducibility are unacceptably low, and that the way to fix the problem is to make a change in perspective where we focus on what we've chosen to ignore as a consequence of what we've chosen to control. So right now, we're all about reporting what we've controlled. We're all about controlling everything that we possibly can. But in fact, that is the fundamental root of the problem in many cases. And secondly, that maybe we need to start viewing animals as patients, not tools. And in many areas, there is also the feeling that we need to act now before it's too late, before we lose too much credibility. And so if you're interested in this, and in 2017, we tried to sort of collect together this literature and put our arms around it and pull out some common themes. This paper basically argues that what we're seeing here is a paradigm shift, a really fundamental change in the way in which people are beginning to do biomedical research. And we work through some questions you can ask yourself to try and improve your reproducibility in translation. But of these, the easiest, the most immediately implementable and most proven solution is experimental design and statistics. And just to illustrate that, it's really nice to look at Glenn Begley's work. So Glenn Begley was involved with onboarding compounds out of academia into a drug company. And this is in cancer. And so his lab basically took a compound. They tried to reproduce the really exciting result that had been published by the academics, and on the basis of that, decided whether or not they were going to bring the compound into the pipeline. And amazingly, he reported about a 10% success rate. What was even cooler about his work, though, was that because of the access that they had to the original research, he was able to go back and look at the original research and try and pick out features which predicted whether or not a academic result would successfully reproduce when he brought it into the company. And it turns out that there are 6 questions he could ask, and if the answer to all of these was yes, then pretty much a compound would be reproducible in his hands. And the first of those is, were experiments blinded? Brianna's going to talk about this in more detail. But just to give you an idea, Kilkenny and colleagues recently surveyed top papers in top journals of biomedical research. And the rates for this are shockingly low, right? So 14% on average of experiments were actually blinded. 13% were properly randomized, again, something Brianna's going to talk about, and bias avoided. And I mean, I'll just be perfectly honest, like if I was grading an undergraduate research project and they didn't blind and they didn't randomize, I would send them away and get them to do it again, right? If they had time. This would be unacceptable. And it really is unacceptable in published work. Another really simple question that he asked was, were the right kind of statistical tests used? And again, Dr. Gaskill's going to work through this in a little bit of detail. But we have some good ideas on that. Nature Medicine, for instance, 2005, had a statistician just flick through every paper in a year's worth of the journal. 38% of them used appropriate statistical tests. This Newenhouse paper I love, and it's looking for a very particular but very common statistical mistake, again, which we're going to talk through later on, which is failing to use interactions in factorial designs when that's the hypothesis under test. He looked through the top 5 journals in neuroscience, found that 50% of behavioral neuroscience papers failed to analyze these kinds of experiments properly, and not one molecular neuroscience paper did. And that's, again, a year's worth of papers out of the top 5 neuroscience journals. So again, this is just simply unacceptable. Like this should be being caught by reviewers. It should be being caught by eye cooks. It's just not okay. And in fact, when we look at all of his 6 questions, were experiments blinded and bias avoided? Were experiments repeated? Are all the data presented? Are there positive and negative controls? Were the right tests used? 5 out of 6 of these are just basic experimental design and statistics. And so the simplest thing we can do is we can go to Glenn Begley's paper, which is only a couple of pages. It's a really nice read. And we can ask that about every protocol that we look at. And I've written a couple of reviews sort of expanding on this a little bit. But right now, I'm going to hand over to Dr. Gaskill, who's going to talk you through how in practice you can think through some of these issues and apply them in your own research, and also look for them whilst you're reviewing papers or reviewing protocols. So thank you very much for your attention. I'll be back for the Q&A, and I'll hand it over to my esteemed and wonderful colleague, Dr. Brianna Gaskill.
Thanks, Joe. So obviously Joe's set a really good example of like what the big picture is, why we're talking about these types of things. With the second half of this presentation, we're really hoping to give you some practical ideas to take back to the lab. And some of these are things that I work through with my graduate students all the time. And so, and are some things that I notice that working with industry that I see over and over again, and some little small things that can easily be fixed in terms of statistics, experimental design, and how you're setting up that experiment that can easily be fixed in order to really help with these reproducibility issues. So before we get started, a lot of what I'm going to be talking about is power and sample size. And like Joe said, power is the change that, assuming there actually is a real world effect, that your experiment will actually detect it. So we want to make sure that the work that we're doing is actually worthwhile. Now, power is actually a function of signal to noise ratio. And I felt like this image really helped represent that if you have a lot of background noise going on, it's really hard for you to detect what that signal is. And so a lot of what people do is attempt to increase their sample size in order to increase the signal. Now, the thing that a lot of people don't realize is that that increase in the signal reduces the noise, but these two things at different rates. And so this can be a little bit challenging in just that being the simple assumption that if you didn't have enough power, you just increase the sample size. And so what we're hoping that by doing that, what people are thinking is that you'll really be able to amplify that signal, reduce that background noise, and really be able to detect the question or test that we're trying to do to really find the true answer. And so we can actually reduce noise through increasing the sample size, but it stabilizes fairly quickly, and really the only way, to a certain degree, and the only way for us to really further decrease that noise is really through good experimental design. So in actuality, any signal will actually become significant if you have a big enough sample size. But this could actually require thousands to hundreds of thousands of data points or animals. And so really what it comes down to is finding that result really worth those animal lives, and is this something that's really going to be translated well into human use. So what happens is that when people increase sample size, this is really a brute force approach, and it's not a sophisticated enough idea, but that really, if we want to reduce that overall noise, we really need to be utilizing those sophisticated experimental designs and having a much more strategic and smart approach to how we're actually going to be running these experiments and really hope to reduce that noise and see our signal in the end. So what Joe and I have done for all of you is to make up a data set. It's completely fictional, based on a generic experimental design. And so what we're going to do is we're actually going to go through together and talk about different aspects of this experiment, how we might actually analyze it in different ways, and what kind of outcomes we can get based simply on same data, same fictional animals, but that we can get different results depending on how you analyze the data. So for our fictional experiment, we're going to have two different main treatments. We have some experimental drug that we're interested in, and of course, we're smart experimenters. We're going to have some type of control group or a placebo. So we would be injecting our animals with one of these two things. But again, we're very smart and we're paying attention to the literature, and some of the other speakers in this lab roots session are going to be talking about the importance of adding both sexes. So hopefully you are already doing this, but we're pretty smart too. We've listened to those talks. We're going to include both of the sexes in each of our groups to make sure that we don't see any interactions that might be different depending on the sex that the drug is being provided to. So what we're going to do, and one of the things that I want to run through with all of you here, because this is going to become important later on when we're going through some calculations, don't worry, they won't be too hard, is to really focus on and make sure that people understand the difference between when somebody reports a big N or a little n in how many animals they may have used. So when somebody reports a big N, this is basically the total number of animals that were used in the experiment. So in our particular example here, we have a big N of 80 because we're adding up all of the animals across these 4 different groups, and that's where we get 80, a big N of 80. Now, if somebody reports a little n of 20, this actually means that in the smallest combination of our drug and sex in a factorial design, so in each of these 4 groups of drug by sex, we have 20 replications, or 20 animals in this case, that is the smallest combination. And so make sure that you understand that when you are reporting in your manuscripts or reports or whatever that might be, that you understand the difference between big N and little n, because that can make a really big difference. And you'll see how it's important to understand the difference as we work through some of these examples. So we have our data set. We're going to start off with probably something that all of you who have ever taken any kind of statistical class, workshop, they've probably walked through a t-test. And this is actually something that we're recommending people not do, but this is usually the first thing that people feel comfortable with. And so with a t-test, we're only going to be testing our treatment variable. So I've run that t-test of our main effective drug. So it's looking at the difference between our drug group as well as our placebo group. And you can see that our probability, our p-value, is not statistically significant. It's 0.12. So we didn't find a difference between our 2 groups based on the main effective drug. Now, something that you want to pay attention to is your R-squared, because this basically tells us how much variation in our data, our outcome variable, were we actually able to explain with our model. So our model in this particular example is just the main effective drug. And we were only able to explain about 3% of the variation in that data. That's pretty bad. So, okay, well, that didn't go as well as we would have liked. And we did not reach significance, which in essence means that you did not reach significant enough power. And so, well, what if we had wanted to achieve 80%? How many animals would we have actually needed in order to reach that 80% power? Well, you can't actually solve for power post hoc. That's kind of a no-no, but we can actually estimate the number of animals that we would have needed in order to reach that level. And then it comes to kind of an ethical question. If we wanted to repeat the study with more animals, would that number really justify the use of animals? And so I went in and I estimated the power based on the parameters in this data set. And we find that we would have needed between 260 and 280 animals in order to achieve that 80% power that Joe was talking about. That is a lot of animals, potentially almost 200 more animals. And I think a lot of us would think that that was probably not worth those animals' lives in order to achieve that power. All right, so that didn't work out. So next we're going to try a two-way ANOVA. And so we designed this into our experiment. So of course we would want to include the main effect of sex because this is something that we were interested in the first place. So of course we'd want to include this in here. So even though we included it, we still are not finding statistical significance in either of these two main effects. So that's not going great still either. We look at our R squared and it has increased slightly, but it's still not very good. This is pretty poor explanation of that variability simply due to these two factors. Again, if we wanted to achieve 80%, how many animals would we have needed? And again, really doesn't change much. We would have needed between 260 and 280 animals. So the t-tests and the two-way ANOVA really aren't giving us that much power. So we'll move on to the next level of experimental designs, and this is called a split plot design. Now don't worry, I'm going to walk you through this. It's actually a pretty relatively commonly used experimental design that was traditionally used in agriculture. So when you say split plot, it pretty much was used for different plots of growing plants. And the thing that's really great about split plot designs is that it helps us deal with real world issues. Let's say that you have a factor that you want to study, but you can't change it as easily as other factors. And so it allows us to kind of work with that. So if we have this general plot of land that we need to test some fertilizers and different types of seeds for, let's say, corn. I'm in Indiana, we're probably growing corn. So we have this plot that we're going to be experimenting with, but one of the things that we need to keep in mind is that not every area of the plot is experiencing the elements in the same way. For some reason, on the top of this plot, that particular area just gets a lot more sun. And at the very bottom of the plot, it's a lot more high moisture content in the soil. And so the top receives more sun, the bottom receives more water content. And so if we want to test some fertilizers, which we can only apply to large sections of land, and we want to try and do this entire study in one growing season because, you know, temperature varies from year to year. And so we want to see how all of these elements can affect these fertilizers and combinations with different types of seeds. So what we need to do is start splitting up this plot into different types of subplots. And like I said, we can only put fertilizer on large portions of our plot. So we could set up our fertilizers with fertilizer A at the top and fertilizer B at the bottom of the plot. But obviously, we're confounding some issues here. We won't know if fertilizer A shows amazing corn growth if that was due to high amounts of sun or low water content, or if fertilizer B works, then it's simply due to low sun and high moisture content. And so we actually can't pull these two things apart and tell what might actually be causing the increases in this corn growth. So we'd probably prefer to set up our plots more like this, so that both fertilizers experience high sun and high moisture content, and we have some nice variation between these two. So the next thing we would want to do is then create some subplots within these two sections of fertilizer that we'll be growing. And this is where we'll put in our different seed varieties. So we have 4 different seed varieties that we're going to try. And what we would do is we would take a random number generator. I'm a big fan of random.org because the random number generator in Excel is not a true random number generator. That we would use a random number generator to figure out which seeds go in which plot. So I did a random number generator and we see this order of seeds. We would use this random number generator again to figure out the order of the seeds that go in fertilizer B subplots. But of course, we'd want to use that, but we would also want to balance across these different subplots. We would want to make sure that, you know, seed 3 didn't always go in the outermost sections or the innermost sections. So we would want to balance across these different subplots. So that's all well and good. We can figure out how to plant and test different types of seeds and fertilizers in Indiana, but how can we apply that to our example experimental design? And what we can do is that if we're looking at this, we need to figure out what that large subplot would be. And so if we're looking at our two main treatments, our drug, as well as looking at different sexes, which of these two things can we not change as easily as the other? Well, most likely that's going to be sex. We can't really change that and assign that to animals based on a random number generator. So really what we're going to be doing is we're going to be separating our two subplots, kind of like we showed in the agricultural example, that we'll have males and we'll have females. And then what we plan to do, so this is our top layer, our between subject layer. And so what we're doing is we're looking at the effect of our drug, of being male, how it's tested and how we can see differences between males and females and between subject. So these smaller subplots are actually our cages. So we have 20 cages for males and 20 cages of females, and we're going to put 2 mice in each cage. So we'll have 1 drugged mouse and 1 placebo mouse. Now, one of the things we also want to make sure that we account for is that we want to blind ourselves. So it would probably be best that I might have a colleague pull up the syringes of either drug or placebo in an animal order, in a cage order for me, so that I actually don't know what I'm injecting these animals with. So this is one type of blinding that we would probably want to initiate here in our sophisticated design. So not only that, but we'd also want to balance from cage to cage which animal actually got the injection first and whether that was a placebo or a drug. So let's just assume that the first animal out of the cage has less time to be stressed due to being scruffed, but the second animal, due to that delay of opening the cage, may actually experience more stress simply because they're sitting there waiting and they know that something's about ready to happen. And so we would want that balanced across both of our different types of drugs. So I set this up just to kind of make that connection back to those plots that we were talking about from the corn example. But this is actually not how we would want to set up these cages on a rack. We would actually want to use more of this checkerboard example where we would go oscillate between males and females. We already have both drugs being represented within each of these subplots. And so maybe we would split these up between rack 1 and rack 2, or perhaps they're on the same rack and on various sides. But one of the things that I run into quite often is that I come into a vivarium and I see that animals' sexes are willy-nilly assigned to a row or column. And so you can see that if we were looking at our males and females here, this is not really achieving that checkerboard design. So we might want to do something a little bit more like this, where we're oscillating back and forth between male and female. But one of the things that we really want to focus on and how this is really like our plots that I was explaining with the corn example is that, let's say this top row, similar to our plot, might be experiencing more sun. Well, obviously in the laboratory, we're not actually experiencing sunlight, but this top row may be experiencing higher light conditions, which has actually been shown to cause stress and affect drug disease progression in different types of disease models. And so we obviously wouldn't want to put all of our treatments on the top row because of that essence of the top row receiving more sun. On top of that, the top row might be experiencing more vibration simply due to those blowers and motors on the top of the rack. And that might vary as you go lower on the rack, on the lower rows. Not only that, but we're interested in the columns as well. So this particular column, as you can see in this image, is closer to the activity that's probably going on a daily basis in the room. People moving things around, people running studies. And we actually know from the literature that in other animals that proximity to the door can actually cause more stress than if the animals aren't as close to the door. So it's possible that these animals in this first column may be experiencing more stress simply due to the activity that might be going on in the room, closeness to the door, than the animals that are closest to the wall. So that's why we're looking at both our columns and our rows and how those things might be affecting the animals, similar to how the water and the sun was affecting our growth of our corn in our plot. So we figured out our split plot design. We're going to now include cage as our subplot. And actually this becomes a blocking factor for anything, any variability that falls within that cage. So it could be light intensity, it could be vibration. The thing that's beautiful about this is that we don't really care. We don't have to know what that is, as long as we can control for how the mice that are in that cage are more similar than the animals in another cage. So we include this blocking factor. And the way that we kind of indicate that we're doing a split plot design is by including a cage factor in our analysis, but we're also going to nest that within sex. And so this is what that looks like in a model. And basically what we're saying is that this cage can only belong to one sex. And so, which makes a lot of sense. It's an all male cage or it's an all female cage. And so this is how we indicate it in the analysis. So let's go through. What did we find? We've got some statistical significance on cage, which actually we don't really care about, but it's telling us that there's a lot of variation from cage to cage in our experiment and between cages within a sex. So this could indicate variability that falls on that cage level. So again, the animals on the top of the rack may be experiencing things that are different than animals on the bottom of the rack. And we can see that from our R squared, we have jumped from 3% with our two-way ANOVA to 96% of the variability. This is humongous in terms of the improvement in this statistical analysis. So if we go back and we look at that power, actually, we probably could have gotten away with even fewer animals than the 80 that we tested, perhaps somewhere around 40-ish animals, in that we could have achieved 80% power between 44 and 48 animals. So we could have gotten away with a lot less animals in this experiment. But we're not done yet. So we have sex in this design and we have drug in this design. Well, most likely the reason we have both of those factors is that we're curious about how those two things interact. So we want to include an interaction of sex with our drug effect. So again, we're just adding this interaction of drug by sex, which you can see at the bottom there. You can see that we're actually improving our statistical significance with sex now being significant. Our R squared, well, we can see that in general, that we are seeing differences between males and females, and that really it's an effect of females. So perhaps there's something to do with how the drug interacts with estrogen within the body that affects the outcome, whatever that is that we're testing, that now we would know that it doesn't really have an effect in males, but it really does in females. And so if we look back at our R squared, it's increased from 96% to 98%. So a small increase, but still fantastic. So in terms of our power, again, we would have needed between 44 and 48 animals in order to achieve 80% power. And remember, we've used 80 animals in this experiment, so we're still way overpowered. So how should we have actually calculated our sample size? So we've already run the experiment, but how should we have started this entire process? Well, calculating power for factorial designs, which is what we did here, is actually really complicated. And you cannot use, I need to stress that, you cannot use a traditional power calculation. That is designed for running t-tests. And we're not running a t-test. We're super smart experimenters, and we are going to be using some of these sophisticated split plot designs. And so, well, how do we do that if we can't use a traditional power calculation? Well, there's something called Mead's resource equation, and it utilizes a curve of diminishing returns that basically shows that as you add more animals, you receive less and less power as you add more animals. And essentially what we're doing with this equation is calculating the number of degrees of freedom for your F-test, so for your model as well as your error. And really what we're looking for here, the really important part, is that the error term needs to be between 10 and 20 degrees of freedom. Now, this is my favorite little thing that I think I've come up with in the last few years, is that Joe and I just published something indicating that we should be using the Price is Right rule when estimating our error degrees of freedom. And basically what this is that you want to design an experiment that brings you as close to 20 without actually going over. Because once after you get over 20 degrees of freedom in your error term, adding more animals really does not provide you with much additional power. So you receive a lot of power up to about 20, but once you go over that, it's not really providing you with much benefit. So we want to find our error term between 10 and 20. So if any of you remember, if you're calculating your ANOVA tables, basically you're calculating the number of degrees of freedom in your model plus what's left over, your error term, and that will actually give you the total amount of degrees of freedom in your experiment. Well, we're interested in error, and so we need to solve for error. So we're going to use our fancy schmancy algebraic information, and we're going to kind of rework this so that we're actually solving for the error degrees of freedom, because that's what we're looking for. We want that to be between 10 and 20. So with our experimental equation here, with our Mead's resource equation, and kind of running through our practice example here, we're going to go through and calculate each of these things. So don't worry, I'm not going to leave you hanging if you can't remember how to calculate degrees of freedom. So the first thing we're going to do is we're going to calculate that total degrees of freedom. Well, that's actually really easy. So it's the total number of data points that you're using in that experiment. For this particular example, we have 80 data points because we have 80 mice. And so you take 80 minus 1, so your total degrees of freedom is 79. So next we'll calculate the degrees of freedom for our model. So we know we're going to be doing that split plot design. So how many degrees of freedom would that cage blocking factor require? Well, this has to do with the number of cages that we have in the experiment. Remember, we had 80 mice. We had 2 mice per cage, so we have 40 cages. And we've nested that within sex. And so we have 2 levels, males and females, for that variable sex. So it's simply 40 minus 2, and that gives us 38 degrees of freedom. The next few are pretty easy. If we're looking at sex, we have 2 levels of sex, male and female. We minus 1, that's 1 degrees of freedom. Similarly with drug, 2 minus 1 gives us 1 degree of freedom. And looking at our interaction, this is slightly different, but we're going to multiply the degrees of freedom for sex by the degrees of freedom for drug. So we've already calculated that both of those are 1, so it's simply 1 times 1 gives us 1 degree of freedom for our interaction. So if we add up all of these for our model degrees of freedom, that's 41 degrees of freedom. So we take 79 minus the 41, so our error term is actually 38 degrees of freedom. And we are way over that 20. We should be between 10 and 20, so we are way overpowered. So we're using more animals than we actually need. Okay, well, that's great. So how would I have calculated this and figured this out before I started the study? So how are we doing our a priori sample size calculations? So our original design, we had 2 different drugs, 2 different sexes, and we had 20 replications. So remember, that's our little n that I was talking about in the beginning. And that gives us a big N of 80. So we had 80 animals in this original design. Well, let's look at maybe having that using Mead's design. So we have 2 drugs times 2 sexes, but in this time, our little n is going to be 10. And so our big N through this calculation is going to be 40. So let's go through and see how this would have worked out with our error term. So we'll walk through this again for our total degrees of freedom. So now we have 40 data points. We have 40 mice in the experiment. So our total degrees of freedom is 39. For our model, the number of cages, remember we're putting 2 mice per cage. So we have 20 cages minus that 2 levels for sex. So that results in 18 degrees of freedom. Simple with the sex, 2 minus 1 is 1. Simple with the drug, 2 minus 1 is 1. And then our interaction, again, really simple, degrees of freedom sex times degrees of freedom drug equals 1. So our total degrees of freedom was 39. We minus the 21, and our error term is 18. So this is perfect. Our error degrees is as close as we can get to 20 without going over. So this is probably a good design using for a factorial for our split plot. So let's actually go in and look at the analysis. So what we did, we used the same data set that I've been using in all these other calculations. I just randomly chose 20 cages, because remember the first set we had 40, and the last set that we cut everything in half, we're gonna have 20 cages. So I just blocked those off and did not use those in the analysis. And we can see that even with half as many animals, we still find statistical significance and have a very high R-squared. So we're really able to identify that variability in our data, in our outcome measure, based on what we're testing in our model. And actually, we possibly could have gotten away with fewer animals. So this is looking at the power for just our main effective drug, and it's actually saying that we could have gotten away with even fewer animals, perhaps around 26, and still achieved 80% power for this experiment. So we reached significance with 40 fewer mice. That is an amazing reduction. So thinking about how we're implementing the 3 Rs, reduction in our animal use, that's amazing. So that's really exciting. We can use this as experimental design. And so we kind of wanted to leave you guys with some things to look for, things to think about as you're designing your experiments and looking at experimental designs that kind of go back to some of these pitfalls to watch out for. So the first of which is confounding treatment with cage. So we might want to reduce animal numbers per cage. So remember, one of the biggest effects that really shot up our R-squared was including that blocking factor of cage. And so we could run this experiment with groups of mice in 4. So our drug group at the top and our placebo group at the top and our drug group in the bottom. But maybe if we actually increase the number of cages and reduce the number of animals, we could actually improve that power by being able to subdivide that variability from cage to cage. The next thing that we could do is we could split treatments across cage. So I've got our experiment here. We've got our drug animals and our placebo animals, but we have one treatment in each cage. So it would be a better design, actually, if we could apply the treatments, both treatments within one cage. So we'd have both drugged and placebo animals, actually very similar to the experimental design we just went through here. Next, we want to make sure that you're not confounding treatment with rack position. And this is where you utilize a Latin square. Now don't worry about that. It basically means you need to do some cage Sudoku. So this is actually a game that I love, and maybe why I love doing this part of experiment design. And so basically, if you don't know what Sudoku is, you get this table and you have to fill in these empty squares with the numbers 1 through 9. Now, the numbers 1 through 9 have to go on a row, but they can't overlap with the numbers that go in the column. And they can't overlap with each of these subsquares, our subplots, perhaps. And so this is essentially what we're trying to do on a rack. So if we line up all of our females on top and all our males on bottom, it's like putting all of our 1s, 1, 1, 1, 1, 1, and then all of our 9s on the bottom. So this is not achieving Sudoku. Well, what happens if we go male, female, male, female? Well, it's the same kind of thing. We have all our 1s on the top, and the next row is all 9s. And so we still are confounding treatment with rack position, or row in this instance. So again, we have not achieved Sudoku. So really what we're looking for is, again, that checkerboard effect, where we see the effects of high light intensity on that top row between males and females across our rows and our columns. Next is failing to blind. So a lot of times people think of, oh, of course we want to blind our data collector. So the person who might actually be writing down or recording different types of our, whatever we're collecting, our main outcome measure. So of course we want to do that. But what a lot of people don't think about is that we need to make sure that we are blinding our care staff as well. So perhaps that person who is providing those injections, making sure they're blind to the drugs being injected, as well as the people who are taking care of those animals. If they know that a particular drug is maybe going to make the animals not feel so well, they might focus all of their observations on or not equally across all of our cages and treatments. And so they might actually bring in veterinary care more often for the animals that are getting the drug. So this is something else that a lot of people don't think about. We need to blind our care staff as well. We can tell them that things are going on, but we don't necessarily want them to know who is receiving which treatment. And then last, sometimes you can't actually completely blind. I do a lot of research where we implement an enrichment, which obviously you can't blind yourself to. You can see it in the cage. And so what we can do is we can actually blind people to our hypothesis. What do we actually think is going to happen? And so if you can't completely blind yourself, you can at least blind people to what your hypothesis is. And this is the reason why. Blinding is so important because we see that in a study, a systematic review run by McCann et al. In 2016, that they found that if you were not blinded, your effect size or the strength of the thing that you're testing was significantly higher if you were not blinded. So this is showing us that even though it's not malicious in what we're trying, or we're trying to fake our data, but subconsciously we're making slight alterations in perhaps some of these outcome measures that's skewing the overall true effect size. Next, we want to make sure that you're analyzing interactions properly. So in our experiment, we were curious about how drug would affect our sexes differently. So obviously, we want to make sure that we actually include that interaction in the analysis. So we wouldn't want to do just the plain split plot, but we would want to include that sex by drug interaction in order to see how males and females differed to receiving that drug. So this is essentially what we would want to include. Next, we would want to watch out for anything justified by a t-test. So if you're sitting on an IACUC and you're looking at someone's numbers justification, you really generally don't have enough information in order to determine whether this was done appropriately or not. Because we never, at least in my experience, I don't see people asking for the overall experimental design or even asking for an example analysis. How are you planning to actually, what statistical tests do you plan on using? And so this is something that I think IACUCs should include in a lot of those proposals so that you can accurately determine whether somebody did a sample size calculation correctly or not. And last pitfall, do not do pilot experiments for numbers justifications. So what we find in the literature is that in general, pilots use very small studies, very few animals, which leads to it being underpowered, which if they do find a significant effect, it's generally going to be an overestimation of the effect size of that particular treatment, which if somebody moves on to do a full-scale experiment, their likelihood of failing is really high. And in the end, we've wasted all of these animals. So we highly recommend not using pilot experiments for numbers justifications, but simply use Mead's resource equation and just do the experiment. So with that, hopefully we've left you with some practical tips and tricks of how to easily implement some small changes in order to improve your experimental design, improve your statistics, and hopefully help with this reproducibility and translation crisis. So thank you to all of the LabRoots organizers. Joe and I have some additional information if you're interested in some of the work that we're doing down below here. And we wanted to point you to two of these papers. He's already mentioned the theory-epistemology paper, but a lot of the things that I just talked about and the topics that Joe talked about were in this Power to the People paper that was published in JLAS in January of this year. So if you want more information, more in-depth explanation, you can find some of that there. And with that, I think we are ready for some questions.
Okay. Hi, everyone. So thank you, Dr. Garner and Dr. Gaskill, for a very interesting and impactful presentation. Like Dr. Gaskill just said, we will now start the live Q&A portion of the webinar. Just as a reminder, if anybody has questions you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many questions as we have time for. So I've got one question here from Virginia. And she asks, is there a way to do a randomized out in the cage? It's very labor intensive to have more than one treatment group in one cage, besides a concern about possible treatment error.
Do you want to take that, Joe, or you want me to?
Yeah. So when Brianna was talking and this question popped up, I highlighted it. Strangely, this is a question that I tend to get asked almost exclusively by cancer researchers. And my understanding, at least from many of my colleagues in cancer research that I've talked to, is that it is very, very usual to have 5 mice in the cage on one treatment, 5 mice in another cage on another treatment, 5 mice in a cage on another treatment. And that's a pretty standard experimental design in the field. The fundamental problem there is that, say you're looking at 4 treatments and you have 5 mice in every cage, people think that their N is 20. It's not, it's 1. Because you have no way of knowing whether your treatment effect is due to the cage or due to the drug that you are giving to all of the animals in the cage. And so the simple answer to that question is, yes, it may be more work for the experimenter, but as Brianna showed with that example data, we went from needing 260 animals to find the drug effect to needing less than 20. And so the time saving, the money saving, the animal lives that are saved, the sunk investment cost that's saved, because instead of thinking there's not an effect there, you actually find it. All of those savings are worth the extra effort of thinking this stuff through. And it is not hard. If you're going to gavage every animal in a cage, it is not hard to gavage one with one compound and another with another. If you're going to do injections, similarly, you've got to inject every mice, just do it. We have a paper that we're writing up right now where I had a grad student doing exactly this with estrogen injections versus vehicle injections. And yes, it was a lot of work, but we did it, and it really broke open the work that we were doing there. That led us to like a major discovery, and especially in behavior work, but also especially in cancer, we really know that the cage is incredibly important, that the cage you're in is actually probably the single largest source of noise in your experiment. And
even in our practice data set, that was really evident that is the biggest factor that that a lot of people are not taking into consideration. And so I understand your hesitance to change up how you're doing in setting up these experiments. But like Joe said, this could potentially lead to you not needing nearly as many animals or nearly as many cages. And so it may come out in the wash in terms of time investment by using some of these more sophisticated designs and applying your treatments across cage or within a cage. So I think it's one of those things that it might be worth trying out. And, the first time you do it, if it's kind of a cluster, try it again, because the more you get used to it, the easier it is. And so that's one of the things that I recommend, is that if you do it once and it just feels so flustered because you're trying something new. And so this is something that I see a lot of groups doing when I've worked with folks in industry, is that they put all treatments on one row and they don't strategize across treatments within one cage. And so I think it's something that's worth trying. And even if it feels flustered the first time, please try it again.
And so,
awesome.
So having been a bit pedantic, and I think both in terms of just going, no, this is just the right thing to do. there are absolutely situations where it's not possible. So for instance, we were doing some experiments where we were administering drugs in the water. And in that case, you are administering drug at the cage level. And so in that case, you can think of the cage as being nested both within sex and drug. And in that case, the smart thing to do is to have as many cages as possible, right? With 2 animals per cage is a much better design if you have to apply the treatment at the cage level. And a lot of the time, though, you can still get into this sort of split plot situation. So imagine you're applying the drug at the cage level, but you're really interested in, you think it's targeting a particular receptor, so you're going to have a knockout and a wild type mouse in each cage now, right? And because you would expect the drug to work in the wild type, but because it doesn't have the receptor to bind to, it's maybe not going to work in the knockout. And so sometimes I totally agree. It is just a lot easier to apply treatments at the cage level. But again, you can get at these powerful designs by maybe then varying something else in the cage, which doesn't require the same level of handling and effort. And honestly, I think if we think about it, that sort of drug by genotype experimental design is actually one of the most common experimental designs we see in biomedical research. And again, that's very easily done by applying the drug at the cage level, having 2 different genotypes in the cage, and then you get back to essentially exactly the same analysis we're talking about. Yeah. And of course, you can layer sex on top of that, and you should.
Okay, moving on to another question. This is from Andrew. Should we instead focus our energy on replacement techniques, e.g. human tissue models, to solve the problems of translatability and reproducibility? Can you address that in brief?
Well, do you want to take that one, Bree?
I'll start, and you can chime in. So I think that's a good idea in order to see if we can start moving some of these replacements forward. But, all of the hot topics with the human on a chip type replacements that are coming through, these are really exciting. But the problem is we really can't use them yet. And so understanding, and even with those humans on a chip, they still don't encompass the complexity and the mental state of the animal. So one of the things that we really can't replicate, and for good or for bad, is that, animals may be experiencing stress due to aspects in their environment. And that stress is altering that overall mechanism of whatever you're trying to study. And so we can't model that on a chip. And so I think that there is some benefit of moving some of these replacements forward. And I will be happy as a clam being completely out of a job, not being able to do anything with animal experimentation. I will do a happy dance on that day. But I really don't think that is going to actually happen in my lifetime. So I think there's some benefits of moving some of those replacements forward, but I don't think they completely model everything that we're trying to get at in the whole animal experience that cannot really be replicated in a replacement. Anything else to add there, Joe?
Yeah, so I have 2 things to add to that. First of all, just to really emphasize the point that Bree made, we know in many of the diseases we really care about, so neurodegenerative diseases, cancer, HIV, we know that in humans, the single biggest predictor of survival and recovery is the quality of your social network. And so, for instance, in 1 year survival in breast cancer, the quality of the social network in the largest study of its kind ever reduces your risk of death 50-fold. That's far greater than most of the compounds we're interested in cancer, right? And so this is actually an active area of research in my lab, is what is the underlying mechanism of these social support phenomena, because they happen in animals too. You can do exactly the same thing in both rat and mouse models in cancer. that actually social housing really changes the underlying physiology of the animals. And so you would never get at that in an in vitro experiment. So number one, this integrated physiology point that Bree made is just incredibly important. And the second is that actually, if you dig around in the literature, you're going to find plenty of people talking about lack of reproducibility in vitro experiments as well. Off the top of my head, I couldn't tell you what those rates are. I was actually having a quick look whilst Bree was talking and couldn't find any really good numbers. But in our own experience, for instance, we see that there is enormous variability from one 96 well plate to another, right? So if you're doing molecular work, you really need to make sure that you have an equal number of case and control samples or treatment samples, whatever it might be, on each 96 well plate if you're having to use multiple for an assay, and you should include the 96 well plate as a blocking factor in your analysis. And in doing that, we've been able to, again, using this R-squared number, the proportion of variance explained, go from very, very poor R-squareds and to really, really high R-squareds. Because actually, the thing that everybody thinks is incredibly reliable because it comes out of a machine that goes ping and automatically gives you a number, just has an enormous amount of variance in it. So all of what we're talking about here applies to molecular assays and molecular work and in vitro work as well.
Great, thank you. Hopefully a little bit simpler one, maybe. From Julie Evans at Nectar, she asks, are you concerned with hormone changes handling both males and females? And if not, why?
Yes. Bree, I think this is more up your alley. Bree, are you muted?
Yes, I was. It's starting to rain and thunder here, so I thought I'd minimize the background noise. So what I was saying was that I personally have not had issues with this. I know a lot of people are mostly worried about handling males and females back and forth simply due to pheromones getting on their gloves and then it causing all of this aggression afterwards. That's actually not something that we have experienced in my lab. Not saying it doesn't exist, but definitely chemical communication is a huge factor that these guys are paying attention to, that we just have no idea what they're experiencing. And one of the things, we're actually studying some of this, at least chemical communication, but within a sex. So we're looking at chemical communication within aggression in mice and trying to figure out, we know a lot about what instigates aggression pheromonally, but are there any things chemical-wise that can reduce aggression? But at least from going back and forth from sexes, I've not experienced that, but it's probably still something to be cautious about. But I think it's more of an issue of not setting up these experimental designs in the right ways that is going to have a bigger effect than just handling males and females back and forth. I've done that a lot myself and not run into any issues. But like I said, it doesn't mean it doesn't exist. I've just not had any experience with that.
Another question from Lord Ventura at AbbVie is, can you also on a rack?
So the short answer there is, again, if I understand this question correctly, it's like, can I put all of my males on one rack and all of my females on another? And then the answer is no, because you don't know whether the effect is due to sex or it's due to rack. So again, that's what you've got to really think about, right? When you're designing an experiment, have I accidentally confounded something I'm interested in with something really uninteresting? Right? And so that's the short answer to that.
to that Sudoku example is that, I was confounding location on the rack with sex in a few of those examples. And so really want to get to that Sudoku, all of your treatment groups across row and each column to make sure that if you have an effect due to light intensity or location in the room or whatever it might be, it's equally distributed across all of your different treatment groups. So I would agree, no, it's not what you should be doing.
Okay. Another question.
I've marked a couple of, okay, there's a couple that popped up that I think are really important. Can I jump in on one?
Yeah, go for it.
Sorry, I don't mean to. So we had one here. Has there been any sort of analysis to show that using any of these parameters or changes in experimental design truly has an effect on reproducibility? And that is from David. Yes. Again, very, very short answer. There are some beautiful large data set retrospective studies, especially coming from people like Malcolm McLeod and Emily Sainer, where they've looked for patterns in the literature. Myself and Hanno Verbal have set up sort of like fake labs and shown that this kind of stuff works. And then there's a really large study that came out recently across multiple labs, which involved Hanno Verbal and Emily Sainer, where they took this to the next level and again showed that reproducibility is massively increased if you just adopt some of these small changes. So yes, there's a very strong evidence base for the stuff that we're talking about today. Sorry, the reason I picked that one out, guys, is I think a lot of the time, a lot of the people in these sorts of meetings, we're almost like preaching to the choir, and people want to do what's right, and they kind of already are ready to make these changes. And the real question is, how do I persuade the PIs? Mm-hmm. And in part of my role here, where I do stats consulting for PIs and for the IACUC at Stanford, there is nothing like showing somebody that they can do the same experiment with 5 or 10 times less animals, and what they thought was a pilot experiment was actually already an overpowered publishable piece of work, right? When people see the amount of time and money you're going to save them with decent experimental design, they're almost always instant converts. There's always some people who'll hold out, but 90% of the time, if you can show them how it hits their bottom line, which is time and money for academics and an industry, it's time and money and success rates, then people are quite easy to win over. And I think that also answers another question which we had here, which is, it sounds like we need a cultural shift. I totally agree. Yeah. The thing that I see, and I think both Brianna and Megan comment on this too, is that cultural shift is already happening in pharma. When I go and work and consult within pharma, I generally see people already beginning to think this way and very eager to implement these things. Where it's not happening is in academia right now, at least in the US. And that's why I think the IACUCs are incredibly important. And I will really emphasize this, because one of the things I've noticed is that the further you get away from a veterinary issue, right, an IACUC is perfectly happy to tell a PI, oh, this analgesia regime is not okay. You have to change your experiment. Or your surgical technique here is going to be problematic. You have to do it a different way. We're totally happy with that. We're totally happy telling an experimenter, we don't care that NIH review group signed off on this. You're still using the wrong anesthetic, and we're going to make you use the right one. We're totally happy doing that. Well, if you actually go and read the OLAW guidelines, chapter and verse, the IACUC is empowered to make any change like that on any aspect of the experiment. And there's actually a question in the OLAW fact, frequently asked questions, is an IACUC allowed to impose changes in experimental design? And the answer is unequivocally yes. So seeing as, and this again hits other questions like improved aseptic technique, improved anesthesia, those are refinements. Improved experimental design is reducing animal numbers. And actually you can also think of it as being a soft replacement, right? And Russell and Birch actually, and if you, for those of us who read it, they talk about improved experimental design where you're reducing animal numbers actually as being a form of soft replacement. Yeah. So I'll toss it back to you guys.
Well, I think not only that, but IACUC's feeling like they have the power to make suggestions like that. But at least with my experience is that a lot of times the IACUC doesn't feel comfortable making statistical suggestions whatsoever. They just don't feel, because every single field does things slightly differently, and they don't feel like they have enough knowledge, and they assume that the PI has sufficient knowledge to know what kind of experimental design they should be utilizing. And I actually see a comment down here from someone at Leiden University, and they said, well, that's why you need a statistician or methodologist on your IACUC team. And that is exactly what I usually recommend, because you need somebody on there who has the strong background of information and understanding about how these things can be so altering in the number of animals that we're using, and ultimately the number of animals that, like Joe was talking about, potentially going through some of these, category E types of experiments, and being ethically okay with that, and making sure that we have strongly justified why you need that many animals. And I really think that that is a really great addition that I'm seeing more and more IACUCs invite statisticians onto their boards, because I think it can do a lot of good.
And so one thing I would add to that is
Go ahead.
Sorry. One thing I would add to that is that really what we're trying to do here is to raise some red flags. And when I do IACUC trainings on numbers and experimental design, that's exactly what I do, right? It's like, I don't expect the IACUC to become experts in experimental design, but if you can just look for a few of these red flags, and when you see them, have a pet statistician. And by the way, the best kind of statistician is at the risk of blowing my own trumpet, somebody like me, who was a biologist first and a statistician second. Because a lot of the time, the people who are pure statisticians, they don't understand like the details of the way the assays work, or the way that or the underlying biology and things like that actually really do influence how you would want to analyze the experiment. So if you can find a pet biologist who does a lot of stats, or a pet psychologist who does a lot of stats, and rope them into your IACUC, that's also a really, really good way of doing this. And by the way, ecologists, field ecologists tend to use a lot of these sorts of techniques. Psychologists tend to use a lot of these kinds of techniques. So even if you can't find somebody in your stats department who's willing to help, you can sort of dig around and say, oh, who's teaching the statistics for psychology course, because every psychology degree program has a compulsory statistics component, and maybe that person would be the person you want to go to.
Yeah, agreed.
Okay, we have a few more minutes. Joe and Brianna, are there any key questions that you guys see that you want to answer? Or shall I just pick one for you?
Just pick one.
Okay. So, do you think of them? Do you want me to do one of the high ones?
Sure.
Okay. So one of the high priority questions is for any experiment, should we use both male and female animals in the same group, or should we use only one of them at a time? Lots of sex questions today.
Well, you should definitely do them at the same time. So generally when I am working with my graduate students, Megan can attest to this, hopefully, that I make them think of all of the combinations of all of their treatments, whether that's the drug, whether that's the sex, whether that's something else that we're interested in. Maybe we're looking at different types of mice or rats. We want to run all of those things at the same time, because what you find is that maybe when you first start off the experiment, you have a heat a heat wave and the air conditioners go out. And so the temperature in the room goes up. And then, later on it gets fixed. And then people are overcooling the building in order to try to make up for this. And so there are all of these small little environmental factors that can affect the overall outcome measure. And so what we want is we want all of our treatment groups, so our drug by sex interactions, to all be affected by these environmental runs. So even though you can't run, let's say, all of those 80 mice at the same time, but you can look at smaller runs over multiple replications of those in order to make sure that whatever those environmental influences are, they're affecting all of your treatment groups at the same time. So I would highly, highly recommend that you're running both sexes at the same time.
Great. We'll do one more. So from Virginia at the Ohio University, she asks of the person doing the experiment per Jeff Mogul's study where he saw differences.
Yeah, that's an awesome question. So Jeff Mogul, by the way, if you guys don't know him, is a really fantastic researcher, and I strongly recommend, if anybody's interested in this stuff, you sort of read back. He has an amazing paper with an old student of his called Chesler, where they went through 8 years of data in the lab, and they simply asked, I'm sorry for the background noise, they simply asked, what predicts pain response? But they're looking at tail flick. And the single biggest influence was the identity of the experimenter. And this fundamentally, I think this is such a good question to end on, because this boils down to everything that we've been saying. Mice live in a different world from us, right? They're crepuscular. They live in a world of touch and smell. They can see colors we can't see. They hear sounds we can't hear. There is no way that you can possibly control everything that matters to a mouse. And it doesn't matter if you have inbred mice that are, like C57 black 6 mice are so inbred they're clones. But in multiple examples, we see that the higher up the rack you go, the more stressed out they are. That stress affects their HPA axis, and that affects their immunology. We've shown that this is an effect in abnormal behavior. We've shown that this is an effect in aggression. There's beautiful work in NOD mice in type 1 diabetes, where again, you see that the mice on the top of the rack are so stressed out that they're immune suppressed, and it takes them much longer to develop type 1 diabetes. So the whole point of this idea of blocking is that we don't know why the blocks are different. We just know that they probably are. And that's the beauty of this blocking and nesting thing, right? Is that we don't have to know why this cage is different from this cage. We just have to know that the animals in this cage are more similar to each other than the animals in this cage. And if you don't actually have a cage effect, which in mice I refuse to believe, but if you don't actually have one, because of the way all of this math works, introducing a block doesn't change the power of your experiment if it is unimportant. Mm-hmm. And so if cage has no effect, it doesn't change any of the math. All of the F ratios stay the same. So you lose nothing by including it. And even if it has a small effect, one that's not actually significant, as far, you would never look at the p-value of a block anyway. It doesn't make sense. But as long as it has some effect, it's going to make your experiment stronger. Mm-hmm. And so the question here was, should we worry about male versus female experimenters? Yes, that's one of the reasons why experimenters are different. This beautiful work from the 60s, but I don't know why people did this, but they looked at the effect of cat feces on fear responses in mice and rats. And if I recall this experiment correctly, and I may be remembering this wrong, so tell me if I'm wrong, Brie. Oh, Megan. They had cats fed regular cat food, cats fed mice, and cats on a vegetarian diet. And the mice were more afraid of the cat poo from cats fed mice over cats on a meat diet over cats on a vegetarian diet.
Yep.
So probably what you ate the night before affects how the animals respond to you. And all of these things can't be controlled. So all you can do is balance them across your treatments. Mm-hmm. And that's the whole idea here. And so whilst we said avoid confounding if you can, you have to avoid confounding. The reason why cage is so powerful is that everything that we think is important to mice sort of clusters on cage, right? It's position in the room, it's position on the rack. These animals may well have that come from the same litter. They're being handled in the same order in the day. Like I once made a list of this and came up with about 20 things that cluster on cage, which matter to mice. So if you have something like the sex of the mouse or, the sex of the experimenter or different technicians working different times of day, as long as you make it consistent and let it cluster on cage and you include cage in your analysis, you don't have to worry about it basically. And the time and the effort that it takes to think these things through, which is a lot, pays off so much. You spend much less time thinking about this stuff and making sure it's controlled for and balanced than the saving in the number of animals that you need and the amount of time that you need and the amount of money you need to do that experiment. And now, of course, there's a whole bunch of new really good questions coming in. So the person interested in ammonia, then Charlie Byrne at the Royal College in London, she actually has a really active project here on cleaning compounds and their impact on mouse behavior and welfare. So I would look her up. Anything to add, Bree?
Nope. Pretty
okay. Well, we will go ahead and wrap up the question section. I will say that we still have all of the questions that were not able to be answered at this time, and we can give our presenters will attempt to answer these and reply to them per the contact information you provided on the on-demand period or during registration. So they will do their best to try to still answer your questions. I'd really Gaskill for their work. And then I'd just like to open up the floor to them if they have any further closing remarks for the audience.
I think the big thing that we've hit home multiple times in the questions as well as the presentation is really looking for those pitfalls. So looking at when you're evaluating your own experimental design, or if you're an IACUC trying to determine whether somebody has done a proper numbers justification, really keeping an eye out on those pitfalls, and in order to help you identify whether you need to ask more questions or you need further justification. So that would be what I would leave the group with.
Anything further, Joe?
Yeah, my two red flags is one, if somebody has figured power using t-tests or the power calculations for t-tests or correlations or regressions, that's actually a red flag, because then we know that they're not actually blocking their experiment. So it's really cruel and unfair on the researchers, actually, right? That the only methods for which you can calculate power ahead of time are the methods you shouldn't be using, which is why we really emphasized the mass balance equation. So that is something that when you see those kinds of power justifications, that probably actually is a red flag. And the other thing that is a huge red flag for me is pilot experiments, which Bree touched on. But just to re-emphasize that point is, if you are underpowered and you get a significant result, that's actually because by accident you saw a larger result than what's true in the world. And so if you then say, okay, this is what I expect my difference to be, you're actually going to underestimate the number of animals you need, and your follow-up experiment is unlikely to be successful. So for reasons that it's not really worth discussing, just but as a statistical fact, if you say I'm going to accept p equals 0.05 as being significant, and you get p equals 0.05, your power is exactly 50%. So if you have a non-significant result, post hoc power is meaningless. Your post hoc power will be insufficient. And if you have a significant result, then your postdoc power will be super high. And we actually saw that in some of the slides that we showed you. So when people are wanting to do pilot experiments, yes, you can have technical pilots. I don't know how to do this injection. I don't know if this method of imaging is going to work. That needs 1 or 2 animals. But pilot experiments to estimate effect sizes are just fundamentally misguided. And there's a beautiful paper by Button, and who's in John Ioannidis' group, that really explains this very, very nicely, and is one of the many papers that we reference actually in the papers that are accompanying this, the review papers that are accompanying this. So if you see something based on t-test, it probably needs to be redesigned. If somebody wants to do a pilot experiment to estimate sample size, that's actually deeply problematic. And I know that's probably breaking a lot of brains right now, but it is a very important point.
Awesome. Well, thank you both again, Dr. Garner and Dr. Gaskill, to all of the participants for attending and for your questions. I hope you all are able to watch more of the presentations today. Have a great day, everyone. Bye.